All right, well, hello everyone, and welcome to the Astrobiology Colloquium. I am delighted to introduce today's speaker, Dr. Jessica Weber from JPL, where she is a research scientist in the Origins and Habitability Lab. She earned her PhD in chemistry from MIT in 2019 with Dr. Tim Jameson and her BS in chemistry from the University of Chicago in 2014. Her graduate school research focused on organometallic catalysis and pharmaceutical synthesis. At JPL, Jessica focuses on applying synthetic chemistry techniques to address questions about the origins of life and habitability of the early Earth and other worlds. She experimentally explores mineral-organic interactions and reactivity related to prebiotic chemistry on ocean and terrestrial worlds. Jessica has been involved in the 2019 Caltech JPL Venus Seminar Series, Enceladus Vent Explorer Eve mission concept, and the EELS Path to Flight Working Group. So please give a warm welcome to Dr. Jessica Weber.
Well, hello, everyone. Thank you so much for coming. I'm really excited to be here to open for AbSciCons. I've been telling people, very excited to see you all next week as well. But today I'll be talking about the photochemical stability and reactivity of prebiotic molecules under Ceres-Like conditions. And so, as most of you are well aware, the keto survey for 2023 to 2032 came out, and a lot of astrobiologists and origin of life people were pretty excited that A, astrobiology was in the title, and B, that there was a whole section, C, on life and habitability, looking at question 9 being on insights from terrestrial life, question 10 being on habitability, and then question 11 being on search for life elsewhere. And so there's a variety of unanswered questions about how life evolved on the early Earth, how this can play into our search for life on other worlds, and addressing this is directly relevant to the decadal survey, which is really exciting for us. And so I'm a chemist by training. And so many of the questions relevant for origin of life and astrobiology are very relevant, are chemistry questions looking at organic materials. And this could be the availability and synthesis of these molecules that are important for biology, amino acids, nucleobases, so on and so forth. This could be the organic reactivity of those products. This could be different ways to take biotic reactions and test them abiotically. And one way, and the way that I answer these questions, is through chemical experimentation. So actually testing this within a lab. And so for this, we need to determine some kind of reactive conditions that are geologically relevant that we can go from. And so in my lab group in the OHL, we do a lot of work with iron minerals. And iron minerals are abundant on a variety of settings on the early Earth and other worlds, looking at Mars, meteorites, so on and so forth. And many of these mineral-rich locations are important for the origin of life. And even outside of that, importantly, we know from work within the group and other work that iron is capable of mediating organic reactivity. And so this is something that we can then use in our reactive systems as either a catalyst or a mediator. And as life is very prevalent on Earth, we need a way to test this abiotically. So we use laboratory settings. A lot of our work is done with anaerobic conditions and an oxygen-free glove box. Like I said, we use iron minerals. And then within this, we can tune our pH, temperature, so on and so forth, in order to replicate the kind of conditions we want, whether it be Mars, ocean worlds, or Ceres, as we'll talk about in a little bit. And understanding where these, how abiotically we can make these organics, their stability, is really important, not just to answer questions about the origin of life, but is important for spaceflight missions. So if we can understand what is abiotically plausible, this could tell us what could be a signature of life, what is unique to life that we don't see in an abiotic world. And then this is important for contextualizing mission data. So on Earth, as I said, life's really taken over all corners of it. So if you go out and scoop up some dirt, chances are the organics you get there are going to be biotic, but we need some kind of standard for what an abiotic world would look like. And this is especially important when you're looking at upcoming missions. So this is looking at New Frontiers, the mid-sized class missions. And this is a mission type where a PI will lead the proposal and pick one of these topics to explore and then propose it to NASA, and NASA will select one, and it'll be very exciting. But on this list, importantly, if you look to the New Frontier 6 and 7, one of the options is Ceres sample return. And so Ceres is, a potentially habitable ocean world within the inner solar system. It's a dwarf planet in the asteroid belt. And there's evidence for liquid water below the crust. And it's been included in the roadmap to ocean worlds, which is very exciting. It's the most water-rich body in the inner solar system. And there is some recent or ongoing geologic activity. And then from the Dawn mission, there have been deposits of organic matter detected. And so with this, it's a high priority for future mission concepts. And just to give you an idea of ice-rich bodies, I really like this figure from one of Julie's papers that kind of maps out the satellites. And so you can see that, Ceres is kind of in this, middle area with a lot of other worlds. So, maybe it's not comparable to Enceladus Europa, but it's still a very interesting body. And since it's so close by, we might as well go. As I said, Ceres was previously investigated by the Dawn mission, which launched in 2007. And I found this very fun figure from NASA that, kind of throws up some stats on the images taken. So it investigated both Vesta and Ceres as a discovery class mission. And this is where we really get a lot of our information on Ceres from. So just as a snapshot, again, probable subsurface liquids, lots of evidence for brine flows. And then it was previously characterized by Dawn. There's some kind of exosphere. And then as far as what we believe could be there, aside from the organics that I mentioned, there's clays, ammoniated minerals, carbonates, water ice, and salt. And so here's just some of the IR spectra from the detections of organics by Dawn. And so you can see both the spectra and then the corresponding geological setting here. So it runs through the organics, the carbonates, and then the ammoniated phyllosilicates. And so overall, this is a pretty exciting area with, organic material, and it's higher than the meteoritic insoluble organic material. There's also some evidence for iron-bearing minerals, and for these, the iron sulfides and iron oxyhydroxides are capable of shielding organics from degradation under radiation, and then these also exhibit reactivity with minerals. And as I said earlier, we know iron is reactive under these spaces and a really important component. So we wanted to set out to better understand how iron-bearing minerals could interact with relevant organics. And so these laboratory experiments allow us to simulate these environments, and under very, very controlled environments, we can test specific ranges, and then we can inform future spacecrafts of likely environment. So this is good for surfaces that are not well characterized. And 2 examples of this that might come up are Titan and Ceres. So there's still a lot of unknowns about how the surfaces look, and we still want to better characterize, where we'd want to take samples from moving forward. So for our objectives, what we wanted to do is to understand how organics are altered after being irradiated under Ceres-like conditions. And we wanted to start with an important prebiotic molecule, sodium pyruvate. We know it is reactive under a variety of conditions. This is something well studied in the OHL. It's an energy source in biochemistry. It does have one aliphatic bond, which is relevant for the detection on Ceres, although really no specific organics were detected. They just had broad functional group detections. And so we wanted to detect this under 2 different conditions, just irradiating the pure organic, which I recognize may not be the most geologically, relevant setting, but we did want to have a baseline for how the organic reacted by itself. And then the organic with a synthetic iron oxyhydroxide mineral. And after this, I will talk about looking at the stability of reactivity of 3 amino acids, glycine, proline, and alanine, under the same irradiation. And for this, we'll test the amino acids by themselves in their pure form, again, to have some kind of baseline. And then for an unreactive mineral, where we look at silica gel, and then lastly, we will test with the iron oxyhydroxide mineral. And amino acids are also very important molecules for biology. And we thought that these were a good mix of of amino acids that did have, structures that were semi-believable for Ceres, but also were, prebiotically relevant, amino acids that life uses. And so just to give you an idea of how these fit into the broader system, the alpha keto acids are often starting materials in our reactions, and via reduction, we can make alpha hydroxy acids, which are important to form oligomers, and there's some evidence that they could be used in, peptide formation, or via reductive amination to form amino acids, which are important biologic molecules. And we've previously shown that these pyruvate, these alpha keto acids can form a reaction network that in an aqueous solution, depending on the conditions you pull, being pH, iron mineral state, you can get basically different results and different ratios of products. And so the ratios depend heavily on the conditions. And one of those things you'll see here on the y-axis is the oxidation state of the metal. And so for the mineral synthesis that I was talking about, we can synthesize minerals at varying iron oxidation within our lab group. And this can be done to replicate a variety of conditions, including a sediment column on Mars, seafloor minerals, so something ocean worlds, and so on and so forth. And then possibly hydrothermal vents, which is a lot of the work Lori Barge does within our group. And so for our experimental setup, we chose to go with our 50-50 iron mineral mix. And this was done in all cases. We thought that this was a representative using both oxidation states, a good middle value, and using previously reported procedures, we mixed the organics with the mineral prior to precipitation. We did this because we wanted the organics to integrate into the mineral structure. And then we precipitated out the iron mineral by raising the pH to greater than 10. After this, we took the entire vial and placed it in a freeze dryer to remove all of the liquid, but maintain basically all of the organic and inorganic material we pulled in. So there were two different experimental setups that we used for this. The first was an initial test that we saw. We just wanted to test reactivity. So what we did is put our desired material into a vacuum sealed cuvette, pulled it on the high vac for an hour, sealed it, and then irradiated it for 24 hours. And like I said, this was only ran in the initial test. We really wanted to get a baseline for what we saw before we built the larger system with a residual gas analyzer. And for this, we irradiated under an active vacuum, and we turned the lamp on and off at different points throughout the experiment to see if the reactivity observed was connected to the irradiation. And for this, we tested a number of analysis methods besides the RGA. We did a bit of Raman spectroscopy, which turned out to be less than useful for this, as after we irradiated the products, we got a lot of fluorescence. Liquid chromatography mass spec, which is very helpful for the materials, which we did not use iron, and then nuclear magnetic resonance or NMR analysis. For our xenon lamp, this is how it compares to the solar flux from the sun on Ceres. And so, it's not a perfect match, but we thought that this was at least getting us close to some of the irradiation. And so here's what the static test looked like. So we had our vacuum sealed cuvette irradiated with the lamp. And after we did this with pure sodium pyruvate, we did see pretty immediately that something had happened. So we saw a pretty distinct color change. This was repeatable under a variety of experiments. And we also saw a change in our NMR. And so for this, the bottom red spectra is the pre-irradiation spectra, and the green spectra above is the post-irradiation spectra. And so, you can see the very large peak at about 2.35 is the parent compound, but we can start to see some more peaks creep in. So we are seeing some reactivity under these conditions, even if a lot of the pyruvate is still maintained. For the active vacuum setup, for this was a much smoother system. We had the vacuum connected, which was very nice, and we could get inline data from the RGA. And so here you can see the lamp off and the lamp on irradiating the sample. And here is a schematic of what it actually, looks like. So we have a number of vacuum pumps connected to this, and the vacuum pumps are sort of below the picture that you can see. And the power supply for the lamp is 2, and then 1 is the main experimental part. And then the third piece highlighted is the RGA. And so for this, then we began to test the experiments on and off, and we tested again, the pure molecule for the sodium pyruvate and the amino acids, and then the molecule along with minerals. And so with the sodium pyruvate, we're very pleased to see that there is an increase of products, specifically CO2, observed when the lamp was turned on. You can see this, pretty clearly with the increase in pressure and mass. And then, kind of going the other way is the sample run. And so when we turn on the lamp, we get an increase in material. When we turn off the lamp, it stops. And then when we turn on the lamp again, we see an increase in material. And for this, we see a similar result with the mineral. So here is an example of sodium pyruvate with the mineral. The 3 runs of this, these are offset for clarity. And then on the right side, you can see a comparison of the lamp on and the lamp off for one of the cases with sodium pyruvate and the mineral. As a note, since we were, doing these on, it's very different when you have 0.1 grams of pure organic material versus 0.1 grams of a mineral mix. And so comparing the amount of CO2 that was lost is pretty typical to do in these standards, but we think that, we suspect that there's, comparable reactivity for the organic present. And here's another comparison between the pure organic sodium pyruvate and the mineral mix. And so interestingly, for the pure as I said, for the pure organic, there's a lot more stuff going on because there's a lot more organic present. But you can see across it that with the lamp on and the lamp off, we do see some pretty clear lines where we have differences in reactivity with the lamp. And so after looking at sodium pyruvate, we wanted to make sure, the pure sodium pyruvate, we wanted to make sure that decarboxylation is what we're observing. And so we purchased 2 isotope labeled standards, where for the middle one, the carbon that would decarboxylate was labeled, and then for the last one, the methyl group carbon was labeled. And so for this, we see an offset for 13 labeled CO2 with the correctly labeled one. And so as we see an increase in product formation during irradiation with this shift, we concluded that, we are getting decarboxylation, and that is what's coming off. We also had, for the pure sodium pyruvate case, we were able to run some mass spec chromatography, and we saw changes after irradiation and an increase in material here. So the red line is going to be the irradiated, and then the black line is the control. And so you can start to see new peaks and new material coming in with the irradiated material. And here are just 2 more examples of new products. And so for this, you can see on the left, the control is the teal line that is completely flat. And then the other products you can see are coming in after irradiation. And again, with the one on the right, this is a new photo product observed at 175 M/Z. And for the control, the second peak is not present. And in the irradiated, we do see some new material. And so we wanted to use the LCMS to really confirm that we were seeing new material and get some more information about what those products were. And so for the Na pyruvate test, we confirmed that it is reactive under Ceres-Like conditions, which is an expected result. There's a variety of photochemistry experiments that have been done in aqueous materials using sodium pyruvate, and it actually is really nice to see that some of this is in line with what we're seeing. A lot of the photo products are similar between the two results. And this occurs with and without the minerals. And one thing we noticed that I haven't touched on until this point is that we do see reactivity occurring during the synthesis. And so we could be, I think in this case, we are irradiating material that isn't just pure pyruvate, it is altering under the conditions with the minerals. And since these are important prebiotic molecules, understanding their availability will tell us what kind of processes could occur on Ceres. And so results indicate that it would undergo decarboxylation. For this, we did see a lot of it maintained, but over extended period of times, we would imagine that this would make the alpha keto acid less available for amino acid synthesis via reductive amination. However, in this reaction, we are breaking down and creating more smaller products. And we think that this is creating smaller reactive molecules that could be, reacted further. And so for this, as I said, at this point, we're transitioning to the amino acids that we had looked at. So we wanted to look at glycine, proline, and alanine. Under these systems, we wanted to apply really what we had learned to look at these. And we thought that these were good examples because, under our conditions, certainly not on a geologic scale, but we expected the amino acids to be at least slightly more stable. And we think that this was a nice offshoot of the alpha keto acids as they're in the same, reaction network. And so looking here, just again, looking back at the reaction network, we had started looking at the starting material of this cycle being pyruvate, and now we wanted to transition to looking at sort of the end product of the reductive amination. And so, again, this is a small part of the larger cycle, but we thought that these were two different classes of molecules that would behave differently under these sets of conditions. And so this is ongoing work, but the RGA spectra does show an increase in products after sample irradiation. And so for this, we're looking at the only the cases with the silica and the iron. And the silica shows quite a bit of what we believe to be is decarboxylation. And the iron also is showing similarly reactivity with the lamp on and off, although we think it's very interesting that the iron mineral seems to have less less CO2, at least as a first pass from this, and kind of less reactivity, because these were both I should also say these were both synthesized the same way as the pyruvate mineral synthesis, where we mixed everything together, solubilized everything, and then crashed out the iron mineral and then freeze dried it. So both of them were made at the same concentration. And so these should be a lot more comparable than, say, the mineral organic to the pure organic, where the organic would have a much higher concentration there. And here is just another way to look at the mineral mixes. And for these, as the mineral is pretty dark, it's hard to visually see any changes, but we do see some, I don't know if you can see on the screen, some slight darkening and discoloring with the mineral, which we thought was very interesting. And we do see the same, effects of this, where we're seeing changes with the lamp on and off, and a lot more reactivity when the lamp is on. And so for the status of these reactions, so we've observed a reactivity in all three cases. And so we are, surprisingly seeing reactivity under these conditions. We're currently working up the LCMS data for non-iron containing reactions. We had some issue with actually exploring the iron containing reactions on the LCMS and removing the iron from it and actually getting concentrations that'll move forward. And so for this, we're working forward at identifying non-volatile material. And if you are interested in this work and attending AppSciCon, a lot of this work was done, or all of this work actually, was done in collaboration with Ellen Sablinski, who's a postdoc at JPL. And this is what she's presenting in her lightning talk at AppSciCon. So she'll have a lot more information than I currently have on it. And, you know, we hope to see you there with our more specific LCMS spectra. And so for this, we've got a number of future directions and things moving forward. And so for this, we really want to expand upon our Ceres relevant parameters. So right now we've done all of this at room temperature, or let's call it room temperature adjacent. As we are irradiating them, we do think there is some variation in the temperature. We started with the iron minerals because they are a reactive subset. But certainly, as I mentioned in the beginning, there are a lot of other minerals on Ceres, including ammoniated material, which could be very interesting, especially when looking at the sodium pyruvate, as well as ice salt mixtures. And a lot of these were, you know, inspired by Dawn, but mostly come from prebiotic chemistry. And so we think going back to Dawn observations could be interesting for some more of the organics. And we also wanted to get visible to near infrared to compare with the Dawn beer, which we thought was interesting. Another thing that we might want to look into is expanding to, let's call it less stable biosignatures. A lot of work that I do is with cofactor material. And so looking at some of these, which we know will fall apart into still large usable materials, could be interesting. And for all of these, we've tested the organics by themselves. And we think that actually, you know, adding some materials like ammonia or multiple amino acids together could really impact the reactivity. And so for this, we've tested 2 different amino acids, and we found two different organic classes, being sodium pyruvate and 3 amino acids. And we found that both were reactive independently and with our synthetic minerals under UV irradiation. There's slight differences, of course, in each case. And this shows that, you know, a variety of organics could be reactive under Ceres-Like conditions. And we think this is especially important for preparing for a future sample return mission concept, to know not only what organics could be present, but, you know, where to look and what we should be looking for. And so as some broad takeaways, we think that laboratory origin of life studies and astrobiology studies are relevant to the planetary and astrobiology decadal survey to understand how life evolved on Earth and could evolve on other worlds. Synthetic chemistry is needed to contextualize mission science and instrument use, and abiotic organic chemistry can be very complex without life. And by understanding these parameters, we can better constrain life detection and mission concepts. And just to, you know, add a few more plugs, I'm going to be at AppSciCon, like a lot of people here. So I have a poster that I'm giving on Thursday on some Enceladus work that I'm doing with Morgan Cable. And then on Friday, I'll be giving a talk that's maybe more in line with previous talks you may have heard from me, which is on iron sulfide mediated quinone reduction to address the origin of metabolism. And then, you know, there's a variety of students and postdocs from our group going, and we hope you'll come say hi to them too. And also just to highlight, again, if you thought this was interesting work and want to hear more about it, more specifics, Ellen will be presenting this on Thursday in the e-lightning hall. And we're pretty excited about her, you know, her talk on this, and we think it's going to be really great. So for acknowledgements, I want to, again, really highlight Ellen's work. We have a paper on this that's in review and hopefully going to be resubmitted soon. And Rob and Brianna have also been extremely helpful with the setup, and Brianna has done a lot of the LCMS analysis. Julie provides a lot of Ceres context, and Lori's, of course, an expert on this mineral synthesis. And all of the NMR was ran by Dave Vanderbilt at Caltech. Ellen was an NPP when she started this project, and this work was funded internally through JPL. And we have a pretty bustling group of students, as I mentioned earlier. So here you can see us, around lab and, hanging out. So I think all of them will be at AbSciCon. And we hope to see you all there. And with that, thank you for your very kind attention. And I would be happy to answer any questions.
Why was glycine, proline, and alanine the only three amino acids chosen?
Yeah, so we wanted to have some point of comparison, and with a lot of published prebiotic literature, glycine and alanine are often utilized. They're the two simplest ones. And we wanted to have one that had a little more of a functional group handle on it, so we added proline. Ideally, we'd really like to expand this work. I think cysteine would be super cool to irradiate for a lot of reasons, including the sulfur group and weird chemistry that could happen. But we kind of wanted to have a very starting point that we could easily understand and move from. And so we thought those were good prebiotic examples, as well as ones that had interesting bonds relevant for Ceres, and ones we could compare kind of along each other.
Did you irradiate all the samples?
No, so we ran two different setups. In the first setup, we irradiated for 24 hours. In the second setup, we radiated on the order of 2 to 3 hours, taking breaks where we turned the lamp on and off at set time points.
And was related to X-rays, this surface.
Yeah, so I'm not going to sit here and pretend that a 2-hour irradiation is comparable to years sitting on Ceres' surface. But we really just wanted to characterize how these materials reacted. So we make no claims that there'll be no pyruvate or not, something like that. But we think that saying that under these set of conditions, we will get decarboxylation, you will get these kind of side products, is a good starting point for understanding if those materials are at all stable, which we're seeing that they're quite reactive. And then, kind of understanding what they break into could help us plan future experiments where there is, propagation. But yeah, unfortunately, we can't irradiate for years.
There's like a lot of advances in chemistry, modeling software. That's when you could like model with software.
I'm sure someone could do it. I'm an experimentalist at heart, and so I would love to hear a collaborator. I would love to have a collaborator that could model something like that and look into it, but that's kind of out of the scope of our lab, but it is an interesting thought.
So when you irradiate the amino acids with the, I guess you're using goethite, the iron oxides, you mentioned new products. You didn't say too much about those products. So can you expand?
Yeah, so with the amino acids, we're still working through what those products are. And we think that for proline, so with the structure of proline, you have that ring within the amino acid. And we think that the ring is breaking off, and we have some ideas that we might get parole or similar compounds coming off from there. And we're also seeing some interesting peaks that could correspond to interesting fragmentation with the alanine that could be related to like formaldehyde or other materials. But we're hoping to better correlate this. So this is what hopefully Ellen's talking about next week, is we're hoping to better correlate this with the LCMS data before we draw any conclusions, and make sure that we're really understanding kind of where we're losing material.
Could it be summarized in essence by saying that you're breaking up?
Yeah, short version, yeah.
And
Yeah.
then presumably the stuff which you actually detect on Ceres today will be, like, and how much do we know about that? Things like acid, something that just, it's very resistant.
Yeah, so I imagine, I imagine there's a lot of stuff we could learn from geologic, organic material, old organic material on Earth. The one thing with Ceres that's interesting is there is a detection of ammonium and ammonium-related materials. And then some of the signatures they found were consistent with aliphatic materials, which could also be, very boring,
Right.
yeah. But that's kind of at least where we're starting from. There were no like distinctive molecules identified.
You mentioned like, mimicking series-like conditions for the radiation, is that just kind of like the amount of radiation? No. Or is that-
Yeah. So yeah, we definitely made some assumptions and stuff there. So when we are talking about Ceres-like conditions, what we're talking about is the irradiation as well as it being under a vacuum.
Gotcha.
So, and of course, like the minerals we have chosen are relevant, and that's that question in the back.
If you could go from the surface to the center. How many changes would you see? Is there a regolith at the surface?
Okay.
And once you wet the rice.
Yeah, so I think from one of Julie's papers, she has a very cool figure or drawing of the internal of Ceres. And so you can see what is proposed here. And so, there's the deep brine mud. And one thing that we weren't able to do, we really would have liked to do like a single layer of material to irradiate, or to have the irradiation material mixed. And within our system, we found that that was really not possible to do without configuring a lot of stuff. That ended up being extremely challenging. We thought it was not going to be challenging, let me tell you. But we think that you can see if I can find it quickly, I'll show you. Okay. You can see that we clearly see some effects of like the very specific irradiation as judged by the burn mark that we found. And it does appear that in our reaction system, it is, when we see that mark, we can see it from the side that it goes, through the material. But I think that one thing that would be really interesting to look at is actually building in layers to this or having it be much more homogenized. So we made no effort for, and this is something we discussed, but we made no effort to sieve or have a specific particle size, which would have been interesting and well controlled. But on the scale of material that we would make, we would have had to really make a lot of material to do that. We would have had to do our sieving inside of a glove box in order to avoid changes in oxidation to our materials. But I think that, I don't know if this exactly answers your question, but I do think that it is interesting to consider what level the irradiation sees, and if there's something we can build in to see kind of how far that would impact and permeate, and at what point we maybe get protection from the irradiation, or at what point we start to really change the reactivity.
That's an excellent point. I had a question about that. So what would you say are the take-home messages for sample return from Ceres? Or what more experiments need to be run to really get there?
Yeah, so I think that one thing that we need to do to better correlate is to get our data into a form that is comparable to Dawn. And so we should definitely run VNIR and analyze these materials to kind of see what the overlap is. Because right now we picked the organics. We made some justification as to why they could be important to Ceres, but a lot of the reason we picked them is because they're prebiotically important molecules that we've seen and used within the lab. And I think that looking at more of a variety of organic material, structurally, from a chemist point of view, I guess there's, maybe not the biggest difference between pyruvate, these alpha keto acids, and the amino acids. And so I think looking at stuff that is much more aliphatic or possibly long-lived, or on the other end, something that we know is very unstable and will fall apart, could tell us what to look for. And I think that this is kind of about like modulating expectation, you know, so it's like we can better understand what we might see if there's, you know, if there's no other processes happening. And again, I completely understand that the time scale is not on geologic time, but at least we have some idea of how these could react.
Do we have any more questions from the audience? Perhaps online? Yeah, I'll just make a quick announcement then. If anyone's interested in joining us for dinner, I believe we're going to Kong Tofu House around 6 p.m. Feel free to let me know or meet us there.